Good morning, students of class 9. We are dealing with the chapter Respiration in Plants. In the last class, we have learned about a few of the experiments on respiration, and today we'll continue with the experiment only. Okay, so today's experiment number 1 will be to prove that heat is evolved or produced during respiration. Okay, so our first aim of this experiment is to prove that heat is evolved during the process of respiration. So for this process or for this experiment, what are the requirements? So we require two thermoflask. We require 30 gram of pea seed or bean seeds, which we have to soak them in water for more than 24 hours. Okay, then we require an antiseptic like carbolic acids in order to prevent the decay or the growth of the bacteria on the dead seeds. Okay, so let's go with the procedure. So what we do, so we will take the two thermoflasks and we will mark them as A and B. We will take 30 gram of pea seeds. We will keep them in water for overnight for 24 hours, let's say. All right, and after that what we do, we will divide the seeds into two equal groups. Let's say our 30 gram of pea seeds, we will divide them into 15 gram and 15 gram. Clear? So kill one group of seeds by boiling them. All right, so what we will do, so we have taken 30 gram of pea seeds or bean seeds. Clear? We have kept them in water 24 hours for overnight, let's say. After that we are dividing it into two equal groups, 15 gram and 15 gram. So one of the group, we will kill it by boiling them. Okay, so 15 gram of pea seeds, we will boil them in order to kill it, and after that we will wash them with dilute formalin, or we will put the antiseptic carbolic acids. You know the function of carbolic acid is to prevent the growth of bacteria or to prevent bacterial decay. Okay, because mainly on the dead seed, there can be bacterial growth if we will not use the carbolic acids, and in that case that bacteria will go for respirations and the whole observations of the experiments will be wrong. Okay, we cannot get the right observations of the experiment. So now one group of seeds has been boiled and we have put the carbolic acids, or we have washed them with dilute formalin in order to prevent the bacterial decay. So after that what we'll do, so in one of the flask, let's say in flask A, we will put the germinating pea seed or the bean seeds. Okay, so let's say in the flask A we are putting live germinating seeds. Clear? And in the flask B we are putting the killed ones. So out of the two thermoflask A and B, in A we are putting the germinating seed and in B we are putting the killed one. Okay, after that what we are doing, you can see over here we are putting the thermometer. Okay, so after that we are putting the thermometer in each flask. So over you can see thermometer, and even this is also the thermometer. Okay, so after that we are putting the thermometer in each flask, and their mouth will be plugged by the help of the wet cotton wool. Okay, so you can see in flask A we are putting the germinating seed, and in flask B we are putting the dead seed with antiseptic like carbolic acid in order to prevent bacterial decay. Then we are putting the thermometer on both the flask, and the mouth will be covered by the help of the wet cotton wool. Okay, so after a few hours what will we observe? Now observation, our observations will be that the thermometer in the flask A will show a higher reading. Okay, so you can look over here. So this is the reading in the flask B, and this is the reading taken after a few hours in the flask A. So when you look at the reading in the thermometer in flask A and flask B after a few hours, you can see that the reading in the flask A has increased. Okay, has generally increased, whereas the reading in the flask B is not so that good. Okay, so only the reading in the flask A has been increased. So this will prove that the germinating seed or the live seeds will give out heat during respiration, whereas the thermometer in the flask B will not show any rise in temperature. So this proves that the germinating seeds will produce heat during the process of respiration. So what we have done, we have taken two flasks We are given them name A and B. We have taken the 30 gram of pea seed or bean seeds. Okay, then we have put them in water for 24 hours so that it can soak the water. After that, we will divide it into two groups, equal groups, let's say 15 gram and 15 gram. So one of the group we will boil it to kill it, and we will put antiseptic, or we will wash it with dilute formalin in order to prevent bacterial decay. So in flask A, we are putting 15 gram of pea seed or bean seed with the germinating seeds. Then in flask B, we are putting the dead seeds with antiseptic. Clear? Then we are putting the thermometer in both the flask, and the mouth of the flask is covered by the help of the cotton wool. Okay, so after few hours, what will we observe? Our observation will be that the reading of the thermometer in the flask A has increased. So there will be a higher reading in the thermometer placed in thermoflask A, whereas there is no rise in temperature in the thermometer placed in flask B. So our conclusion will be that the germinating seeds will give out heat during the process of respiration. Clear? Okay, now next experiment of the day. To prove that, or to demonstrate anaerobic respiration. So now we are going to prove the process of anaerobic respiration. That means respiration, those that occur in the absence of oxygen. Okay, so what we do, we will take 8 to 10 soaked and peeled pea seeds. So we are taking the pea seeds which have been kept in water for overnight or for 24 hours, and we'll peel the pea seeds in a test tube. Okay, so that means, look over here. So this is the test tube which you can see over here. This is the test tube, and inside the test tube we are putting the peeled and soaked pea seeds. Clear? Okay, then invert the test tube in a beaker filled with mercury. Okay, so invert the test tube in a beaker filled with mercury. So the seeds will be completely surrounded by mercury. So now what we are doing, so in a test tube we have taken the pea seeds. Now we are inverting the test tube in a beaker containing mercury. This is the beaker. Okay, this is the beaker, and this one is the test tube, and this is the seeds. Okay, so after putting the seeds in the test tube, we are inverting it in a beaker containing mercury. So this will lead to the filling up of the test tube containing seeds with mercury. So you can see that the test tubes containing the seeds has been completely filled up by the mercury. Okay, now next one. So after about 2 days, the level of mercury in the test tube will fall. So after a few days, what we'll observe, after 2 to 3 days, what we'll observe is that the level of mercury has been decreased in the test tube. Alright, so after 2 to 3 days, we'll observe that the level of mercury in the test tube has been decreased. Clear? Now, the test tube will fall, okay, and the liberated gas will be found to be carbon dioxide. First, let's check it out. Carbon dioxide can be tested by introducing a stick of potassium hydroxide into the test tube, which will float up through mercury, and on coming into contact with the gas will absorb the carbon dioxide, and the level of mercury will again rise. So what we are doing is that we are saying that after 2 to 3 days, the level of mercury in the test tube will decrease. Clear? So the gas which is being produced over here is mainly carbon dioxide. So due to the production of carbon dioxide, the level of mercury has gone down inside the test tube. Now this gas, carbon dioxide, can be tested by putting a stick of potassium hydroxide into the test tube. So what we are doing is that now we are putting potassium hydroxide into the test tube. Now potassium hydroxide will float through this mercury, and when it comes in contact with carbon dioxide, all right, it will absorb carbon dioxide. One more time saying that the function of carbon dioxide is to absorb the function of potassium hydroxide is to absorb the carbon dioxide present inside the test tube. So when we are putting the stick of potassium hydroxide in the test tube, it will float through the mercury, and when it comes in contact with carbon dioxide, it will absorb it. Okay, so now what happens? So inside the test tube there is carbon dioxide. Now it has been absorbed by potassium hydroxide. So when the carbon dioxide has been absorbed by the potassium hydroxide, the level of mercury, look over here, the level of mercury will again rise. Okay, so when this carbon dioxide has been absorbed by potassium hydroxide, again the level of mercury which was fallen in this test tube will again rise. Okay, so for the control setup, what we can do is that mainly for the control setup, because we have got two type of setup, experimental setup and the control setup. So this was the experimental setup. So this experiment can be proved by taking the control setup also. Mainly in the control setup, what we have to do, we have to simply put the date sheet or the boiled sheet along with the antiseptic. Okay, in that case no gas will be produced in the control setup. So what we are doing is that we are trying to prove the process of anaerobic respiration. And what are we doing? So one more time I will explain it so that it will be clear for you. So we are taking a pea sheet, okay, which has been soaked in water and which has been peeled and which has been put in the test tube. This is the test tube. Clear? Now we are inverting this test tube into a beaker containing mercury. So when we are inverting the test tube in a beaker containing mercury, the test tube will be filled up by mercury. You can see in the figure. Now after 2 to 3 days, what will we observe? We will observe that the level of mercury in the test tube has been decreased. As you can see over here, after 2 to 3 days, the level of mercury in the test tube has been decreased, and the gas which is being produced inside the test tube is simply carbon dioxide. So what we can do now, we have to introduce a stick of potassium hydroxide. Okay, that potassium hydroxide will float through the mercury and will go and react with or come in contact with carbon dioxide. When KOH or potassium hydroxide comes in contact with carbon dioxide, it will absorb the carbon dioxide. So when carbon dioxide will be absorbed by potassium hydroxide, the level of mercury in the test tube will again increase. So it will completely surround the test tube. Okay, so for the control setup, what we can do? For the control setup, instead of putting the germinating seed inside the test tube, we have to put the dead seed or the boiled seed along with antiseptic like carbolic acid. In that case, in the control setup, no gas will be produced or will be liberated. Now precaution to be taken. Peeling of the seeds coat of the germinating seeds before introducing them into the test tube may hasten the outward diffusion of carbon dioxide. So this thing has to be taken care into consideration. Okay, so here we have finished the experiment on respiration. Clear? The answers for the progress check, I will supply it through your WhatsApp group. Clear? Okay, now next one. Respiration contrasted with photosynthesis, or we can say difference between these two processes. Now mostly the process of respiration and photosynthesis is usually different. Clear? We have mentioned that photosynthesis is an anabolic process, whereas respiration is an catabolic process, that we know. Okay, so these two processes are completely opposite with one another. Why? Look over here. The requirement of one are the product of the other, and therefore they are complementary to each other. That means the end product of one of the process is the requirement for the other process that we will discuss. Okay, so for that let's go through this table. Number 7.2, photosynthesis occurs in the presence of chlorophyll, that we know. All right, respiration occurs in all the living cells. Photosynthesis occurs in presence of light. Without light there will be no photosynthesis, but respiration occurs at all the times. Photosynthesis uses carbon dioxide and water. Respiration uses oxygen and glucose. So here you can see that during respiration carbon dioxide will be produced, and that carbon dioxide given out during respiration is being used for the process of photosynthesis. So you can see over here, during photosynthesis oxygen is released as the byproduct or the end product, and that oxygen produced during photosynthesis is being utilized for the process of respiration. So carbon dioxide is released is the end product. So this carbon dioxide is being used during the process of photosynthesis, and oxygen produced during photosynthesis is being used for the process of respiration. Clear? Number 5, light energy is converted into chemical energy, whereas over here chemical energy is converted into heat and partly into energy. Results is gain in weight, but in respiration there is loss in weight. In photosynthesis, food or glucose is produced, but in respiration, those food particles which are produced are broken down. Alright, they are oxidized to produce energy. So that's why photosynthesis is known as anabolic process, because there will be production of food, whereas in case respiration is called catabolic process because there is breakdown of food. So there is the difference between respiration and photosynthesis. Now let's compare the process of respiration in plants and animals. In plants there is no gaseous transport. All right, the gases will simply diffuse through cell to cell diffusion. So in plants there is no such gaseous transport like in case of animals. In animals the blood will transport the respiratory gases, but in plants there is no such chemical or fluid which will help in the transport of gases. So in plants there will be cell to cell diffusion of gases. One of the end products of anaerobic respiration in plants is ethanol or ethyl alcohol. So the end product of anaerobic respiration in plants is ethanol or ethyl alcohol. And in case of animal, the end product of anaerobic respiration is lactic acid. Okay. In plant, the process produces little heat as compared to the animal. So there is the difference between respiration in plants and animals. Clear? So in plants there will be cell to cell diffusion of gases. In animal is done by the help of the blood. In case of plants, the end product of anaerobic respiration is ethanol or ethyl alcohol, whereas in case of animal, it is lactic acid. In plants, little amount of heat will be produced as compared to the animals. So here we have finished the chapter. The answers for your progress checks and the intake questions will be supplied to you through your WhatsApp group. Okay. So in the next class, we will begin with the new chapter.